You got some FASTA files from a colleague and wonder what they are? Well, you came to the right place. Hello, and welcome back to our channel. Today, we're delving into the world of bioinformatics to uncover the secrets of the FASTA file format. First off, what exactly is a FASTA file? Well, imagine it as a digital blueprint containing genetic or protein sequence information. It's widely used in bioinformatics for storing and exchanging biological sequence data. Let us look at some real examples of sequences in a FASTA file. In the first example, we see the RNA sequence of a gene transcript. Although we are specifically looking at a gene transcript here, there is no limit to what we can represent as the sequence. It can be a few nucleotides or amino acids all the way to a full chromosome or even a full genome, the latter in the case of most prokaryotes. In the second example, we see the same gene transcript but now translated to a proper protein, including the translating stop, which is optionally indicated by an asterisk. The first two examples were generated via a bioinformatics workflow where we sequenced a new species. The third example is what you would commonly see if you download FASTA files from websites such as the NCBI or the EBI, or from special sequence databases. You can see that the annotation, or description, indicates that this is a sorrel synthase messenger RNA from Amimagus, also known as bishop's weed. In the last example, we see a part of a sequence where the accession, or the identifier, is made up of multiple elements. Now that we have seen some real-life examples, let's break down the structure of a FASTA file in more detail, starting with a very simple overview. Each entry typically begins with a header line, denoted by a greater than symbol followed directly by a unique identifier and optional description. The description must be separated from the identifier by a white space. The sequence data itself, nucleotides or amino acids, comes next, spanning one or more lines. Blank lines at the end of a sequence are possible, but not common anymore. Our first slide here is one of the most simple and minimized FASTA structures, where we just have the number 1 as our unique identifier, and the string of adenines, thymines, cytosines, and guanines as our sequence. The next example slightly expands on this, and we have added a description of this simple sequence. If we have a closer look at this sequence, we see that an identifier as you would commonly see them in FASTA files, an accession made up of letters and numbers, and in this case we have an added version number, separated by the period, as well. Everything before the white space should be unique in a FASTA file. The annotation, or description, following the identifier is free text and it contains as little or as much information as needed, with the one caveat that in the current FASTA format, they cannot span multiple lines anymore. That means, no new line characters except for at the end of the description. Some FASTA entries seem to have a more complicated structure, but remember that everything after the greater than sign, and before a white space, is considered the unique identifier. In some cases, such as older files downloaded from NCBI, we see an identifier that has several elements separated by a bar character and might contain accessions for different databases which can be extracted individually by bioinformatics workflows, but the full identifier is unique. What we are seeing here is another example where the sequence doesn't have a special name, but is indicated to be a segment of chromosome 6 with start and stop locations included. As we see, there is a wide variety of how the identifier can present itself, and in the amount and type of description that is available. One more thing, you have heard me speaking about unique identifiers. Why unique? Well, most FASTA files you will encounter will have multiple entries, tens to even millions of entries. Bioinformatics tools that deal with these files need to be able to pick out, if necessary, a specific sequence, and that can only be achieved if the identifier is unique within that file. Incidentally, FASTA files that have more than one sequence entry are also called multi-FASTA files, but often that distinction is not made explicitly. We have focused on the header line for quite a bit, but of course, the actual sequence is just as important. As we mentioned before, the sequence can consist of one long line, or, more often, multiple lines that are formatted to be anywhere between 60 and 80 characters long. The latter was done, historically, for two reasons. Older tools were requesting memory in certain chunks when our computer memory was sparse and needed to be able to fit this in. Secondly, older computer monitors were limited to the number of characters they could display on one line. We have grown out of that era, but we still see a lot of residual effects even in our bioinformatics tools and files. We mentioned that the sequence lines can contain either nucleotides, that is, RNA and DNA, or amino acids for peptides and proteins. On this slide is indicated the alphabet you would encounter for both of these types of sequence data. In the case of DNA and RNA, the ever-abundant letters you will find are A, C, T, and G. Some entries might have an N character, which indicates any base. Often that actually means, we don't know. The U character will be present in case the sequence is RNA-centric, as the thymine is replaced by the uracil. However, RNA, such as gene transcripts, are most commonly listed with their DNA-centric nucleotide T. All other letters you might encounter are for specialized cases, for example, DNA or RNA profiles where we have to indicate that different bases are possible on that location. On the other side, the amino acids are more straightforward as we will have an almost full one-letter-to-one amino acid mapping. With, of course, the always present exception of the X character, which can indicate any amino acid. Stop codons, or translation stops, are optionally indicated by an asterisk. We had a good look at the content of FASTA files. Now, how do I know if a file is a FASTA file if I am just looking at my folder or directory? Well, most generic FASTA files have an extension of .fa, .fas, or .fasta. The other common extensions are .fna and .faa, which are DNA or RNA sequences, which can be genomic or amino acid FASTA files, respectively. The other lesser common extensions are .ffn and .frn, which are for either protein coding regions or RNA coding regions of genomic segments. In addition to the extensions we mentioned, FASTA files can be compressed to reduce their often large size. If you see an additional .gz or .bzip2, then that means they have been compressed by gzip or bzip2. Now that you learned about the structure of FASTA files, here is a tidbit of interesting facts about the history of FASTA. Both the history of the bioinformatics software FASTA and its format are intricately linked with the collaborative efforts of William Pearson and David Lippman. In 1985, while Pearson was at the University of Virginia, he began developing FASTA as a means to search sequence databases efficiently. Working closely with David Lippman, then at the National Center for Biotechnology Information, NCBI, the duo combined their expertise in bioinformatics to refine the algorithm and enhance its usability. Together, Pearson and Lippman refined the FASTA algorithm, pioneering a heuristic approach that enabled rapid identification of local sequence similarities. This breakthrough allowed researchers to perform comprehensive sequence comparisons in large databases with unprecedented speed and accuracy. As a result, FASTA quickly became a cornerstone tool in bioinformatics, empowering scientists to analyze and interpret biological sequences with greater efficiency. Additionally, the accompanying FASTA format, which they developed, provided a standardized means to represent biological sequences in text files, further facilitating the exchange of sequence information across different platforms and databases. Over the years, Pearson and Lippman's collaborative efforts continued to evolve FASTA, adapting it to meet the growing demands of the bioinformatics community. Their pioneering work not only revolutionized sequence analysis, but also laid the groundwork for subsequent advancements in genomics and computational biology. Today, FASTA, and specifically the FASTA format, remains a fundamental cornerstone in bioinformatics, reflecting the enduring legacy of Pearson and Lippman's partnership in shaping the landscape of sequence analysis. In the present day, FASTA files continue to play a crucial role in life and medical sciences, serving as a fundamental format for storing and exchanging biological sequence data. These files are extensively utilized across various domains, facilitating a wide array of analyses and applications. In genomics and genetics, FASTA files are indispensable for storing nucleotide and protein sequences obtained from DNA sequencing experiments. These sequences serve as the foundation for numerous downstream analyses, including sequence alignment, variant calling, and genome assembly. Moreover, FASTA files are utilized in comparative genomics to identify evolutionary relationships among species, annotate genes, and study genomic variations associated with diseases. In medical research and diagnostics, FASTA files are pivotal for analyzing genetic data relevant to human health. Researchers and clinicians rely on these files to store sequences of genes associated with hereditary disorders, cancer biomarkers, and infectious agents. By comparing patient-derived sequences to reference genomes stored in FASTA format, scientists can identify disease-causing mutations, predict drug responses, and develop personalized treatment strategies. Furthermore, FASTA files are integral to bioinformatics pipelines and software tools used in life and medical sciences. Many bioinformatics algorithms and databases accept FASTA-formatted input, enabling seamless integration into analytical workflows. This interoperability ensures that researchers and clinicians can leverage existing tools and resources to extract valuable insights from biological sequence data efficiently. Overall, the widespread adoption of FASTA files underscores their enduring significance in life and medical sciences. As the volume and complexity of biological data continue to grow, FASTA files will remain indispensable for organizing, analyzing, and interpreting sequence information critical to advancing our understanding of biology and improving human health. Today, we delved into the structure of FASTA files and how they are being used in the life science and medical fields. FASTA files have revolutionized the way we store, exchange, and analyze biological sequence data. If you found the presentation insightful and wish to explore more topics in bioinformatics, genomics, or any other related field, feel free to subscribe for more videos or ask for further assistance. Let me know in the comments how you use FASTA files in your field of research. Stay curious and keep exploring the fascinating world of science and technology.